Next speaker is Dr. Greg Cote, medical oncologist from Mass General, who works in the Chordoma Center here in Boston at Mass General. And he's going to talk about the role of clinical trials and personalized therapy for chordoma patients.
Okay, great. Thanks so much for the introduction. It's nice to see a lot of familiar faces. So I'm one of the medical oncologists here at Mass General. I wear a couple different hats, but I take care of patients in our sarcoma group and see chordoma patients in that context. And then the rest of my time, I run clinical trials and write clinical trials within the sarcoma group and also within our phase 1 group, which is these first-in-human studies that I'll talk a little bit about today. So I was asked by the team to give you a little bit of an introduction into how clinical trials are run and then talk about this concept of personalized medicine, which I'm sure you've heard about, and we'll delve into that into some detail. So this is just a basic outline. We'll talk about, as I said, about clinical trials. I do want to, you've heard a little bit about cell biology from some of the other speakers. I do want to go back to that to revisit it because I think it's really key to understand that, to know what we're talking about when we say this term personalized medicine. So we'll go back into that. And then I'll spend a few minutes talking about how I think about connecting chordoma patients or other cancer patients to clinical trials, and we'll go through that. So this goes without saying, we obviously need new therapies for chordoma. We think we're getting closer to managing the local problem, but there's a lot of morbidity with the surgery, with radiation, and then for those patients who have recurrences or where it's spread to other areas, we're really not making a lot of progress. So you can conceptualize clinical trials as just ways that we think of testing new therapies to try to address this problem. So in simplest terms, this all starts in the lab, and we heard about that from some of our colleagues earlier. This could be down the street at the Broad Institute, some of the biotech industries around the corner, or in academic labs at our hospital, or coming from big pharma. But it all comes from some pathway that a scientist, a pharmaceutical company thinks is important for cancer. They develop a molecule to try to block that pathway and look at it in mice and cell lines. And it may look great in a mouse, it may look great in cell lines, but you really don't know if it's going to work until you get into people, and that's where clinical trials come in. So the very first step is this thing called phase 1, or first in human, and this is the very first time that we give this compound to a person. Now, as I said, it may look safe in a mouse, may work great, but you really don't know, is that dose going to translate the same to a person? Is that safety going to translate the same? So we have to do these very careful, very controlled studies where we explore that dose finding and safety analysis. Now, as part of that, we're also hoping that we're starting to see some signs of clinical activity to give us some clues where to explore this drug, and we'll talk about that as we go through. So this is just a pretty simple cartoon here of how we run these trials. So on the bottom, these just represent 3 patients that we treat at some predetermined dose level before starting the trial. So some very low dose level that we think is going to be safe. We treat just 3 patients. So we may have 10 centers open across the world for this study, but treat only 3 patients across the world at one given time. They can be all types of cancer. It doesn't have to be chordoma. It doesn't have to be breast cancer. It could be some mix of different patients. And we follow them for some time. So we watch them closely, we do some scientific tests on them, make sure they're safe. If that goes well, then we increase the dose, treat 3 more. If that goes well, we increase the dose, treat 3 more. And eventually we keep going. We start to learn what the side effects are and what the right dose is for this drug. And of course, along the way, we're hoping to see that this is working as well. Once we've figured out what we think is the right dose, we open the study to something called dose expansion. There's a lot of nuance to this, but in general, you can think of it as treating more patients at the dose that we think is right. It may be focused to certain diseases that we see the signal. It may be open to all comers. So once we've figured out what that dose is, we want to expand this to more specific populations of patients, and that's where phase 2 comes in. And there's some blurring of activity between phase 1 and phase 2, but the general concept here is trying to see some hint that this is active within a disease of interest. So now, rather than looking at individual patients and increasing the dose, we're taking all the patients, hopefully the same disease. Sometimes it's the same, different diseases, but the same mutation, and we're treating those patients with that drug. Everyone gets the drug. There's no placebo, at least with this model. There's some variations, and we're hoping to see some sign that this drug is better than what we would expect in other patients. So maybe it's time to progression, maybe it's the number of patients that have their tumor shrink. The idea there is that you're seeing some sign that says this drug is good, this drug is worth moving forward into bigger studies. So once we get through that, we come to these big phase 3 studies, and these are the ones that you hear press releases about on the news. And this is where you're trying to say, is it better than some standard of care that's pre-existing? Is it better than placebo? Very hard to do these studies in Chordoma, but for more common cancers, the idea is comparing your new drug A to placebo, comparing it to some standard of care. This is usually a randomized study where there's some kind of coin flip mechanism that pulls one patient into the other study, and you're looking here for clear evidence that it's better than the other. So why would we do these studies? Why would we do randomized studies? Well, even if we see really exciting activity in phase 2, there's a lot of what we call bias, selection bias, and patient bias in those studies, because typically the patients we tend to treat in those studies may be more fit than the general population. So it really takes a nice, carefully controlled study in phase 3 to really figure out, is your signal real or is it some anomaly based on your patient selection. So this is a lot of work, and this is not something that happens within a year, this process moving from one step to the next. And these numbers are a little dated, but at least in the last decade, it was estimated it would take about 10 years to develop a new drug and $1 billion, which is pretty astounding numbers if you think about it. This is part of what's driving the high cost of cancer therapies, and you hear about this in the news, and it's a hot topic during politics, but it's really not a practical model to take a drug through this development process and with the modern considerations with healthcare costs. So we're trying to get smarter about this, and there's a huge amount of effort within academic institutions, within pharma, to try to figure out ways to be smarter about this process. So part of that is this concept of personalizing medicine for patients. How can we figure out which pills, which drugs to give to which patients? And this is where all this genetic testing that you heard about comes into play. We're trying to be smarter about directing patients to trials. And then tied to that, if we have a new drug and it's really not showing activity by the time we're getting out of phase 1 into phase 2, we really adopted an approach of trying to kill that project early, not going all the way through phase 3, wasting all that time, all that effort, all those patients who participated for something that's going to fail. So we're trying to get better at that. And I think that's been a real key innovation to our drug development community in the last 5 years or so. Obviously, this is a unique challenge for rare tumors. Part of sarcoma is seeing uncommon tumors. There's probably at least 100 different types of sarcoma. Some of these I might see 1 or 2 cases a year. Very hard to do a randomized phase 3 study with several hundred patients in that population. So we have to be a little bit creative and how we approach that. And Josh talked about that earlier, sort of latching on to some existing drug development strategies is one approach to that. Okay, so let me take a little bit of a detour here, and I want to talk about some basic cancer cell biology. And I'm just you heard a little bit about this with some of the earlier speakers, but I think it's worth revisiting again. So in a given person, we're made up of cells, and it's hard to know that without looking under a microscope, but there's trillions of cells in your body, and they have this membrane here that you can see, and within that there is this nucleus that Dr. Flanagan was talking about, and inside there is packed in all 20,000 genes of your body, all compacted down together. And those genes are made up of this thing called DNA, which I'm sure you've heard about. So DNA, when it's ready to be used as the code to develop something, is unwound from that tight packing and transcribed into this molecule called RNA, which is then translated into proteins. And the proteins are what goes about and does all the activities within a cell. So the numbers these are just estimates here. That's a typo, it should be 20,000. But there's 20,000 genes in the body, and estimates are in the trillions of cells. And those cells are not just sitting there. They're actually constantly turning over. They're constantly being maintained. So they have to go through this very tightly regulated process called a cell cycle, where within a matter of minutes, they can actually copy all their DNA, all 30,000 genes, and then divide and to make a copy of itself, which is pretty remarkable that this is even feasible. This is a cartoon I stole off the internet that shows just one of these cells dividing over time. So you can see it copies its DNA and then splits. It's pretty remarkable. This can happen. So this is actually an error-prone process, and mistakes happen. It's just, I mean, just technically thinking about making 30,000 copies, you're going to make errors along the way. We have built in this huge network of DNA repair mechanisms that pick up these mistakes and try to find ways to repair them. And if that malfunctions, there's actually brakes on this system that will stop the cell cycle from going, stop it in its place so those cells can't grow because they're abnormal. They have made mistakes. And there are actually built-in programmed cell death pathways that if a cell is stuck here, if it's some mistake, that cell will actually commit suicide and kill itself, which is, saves us from those developing into cancer. So it's estimated that there are billions of cell divisions in your body per day, and anywhere from 1,000 to 1 million mistakes per cell per day. So it's a huge number that are out there, and it's pretty remarkable that cancer doesn't happen more frequently than it does. So how does this translate to a cancer developing? Well, I think of this in a couple of different ways. So one is that one of these mistakes happens, and it happens to be in some critical area, and it escapes this process. It doesn't get repaired the correct way. It gets through these breakpoints on the cell cycle, and that leads to some pathway addiction that drives this cell to make it grow when it shouldn't grow and survive when it shouldn't survive. Another model is that many different small mutations will accumulate over time, and together those will lead to cancer developing. And what we've learned is that almost universal to all cancers is these features where they resist this process of programmed cell death. They have uncontrolled cell growth, so they get through the cell cycle. They can avoid the immune surveillance, as you heard about from Dr. Hiri. They can recruit abnormal blood vessels. They can activate invasion programs where they travel to other areas. And then, one area that I'm interested in that I do research in is this concept of genomic instability. I don't know if this is true for chordoma yet, but the idea is that you already have a mistake-prone DNA repair process. As this cancer goes along, it starts making more and more mistakes, leading to more and more mutations, perhaps escalating its aggressiveness over time. So, when I'm not, hanging out with my kids or seeing patients, I'm thinking about ways to kill cancer cells. And when we as a community think about targets for tumors, we think about all of these processes, and there are any number of different pathways where we're developing drugs. So some of these are novel chemotherapy agents, so these are poisons, but the idea is to kill more of the cancer cells. Can we overcome some of these processes of uncontrolled growth? Can we take old chemotherapies and find new ways to deliver them? So this could be repackaging them into some container that can get into the tumor better. It can be linking it to an engineered antibody that will bind to the tumor. We have many different therapies that look for specific mutations for which we can develop drugs for. We don't know if this is important for chordoma yet, but for diseases like breast cancer, melanoma, lung cancer, it's very well established that this concept works. We heard from Dr. Hiri about immunotherapy. There's a number of drugs that were developed in the last decade that target abnormal blood vessels. I think this is a little less exciting than it was even 5 years ago, but nevertheless, it probably still plays some role that we're still working on. And then, as I mentioned, there's this concept of genomic instability. And the idea here is if you already have a tumor that's somewhat genomically unstable, so has difficult time repairing those mistakes, if we block the existing repair pathways, can we cause so many mistakes that it's catastrophic that those cells die? It's kind of a neat concept. All right, and then, okay, so let's come back to personalized medicine and genetic testing. So we can take a group of chordoma patients or any cancer patients represented by the line of people there, and we can look at their DNA. So we can do this today if we wanted to, and this could be off of a fresh biopsy where somebody has a new biopsy through our radiologist, or it could be taking some tumor that's stored in pathology and we pull it from the slides, and we can actually analyze these and now divide this group of patients into different cohorts based on the mutations that we find. Not every patient's going to have a mutation that we detect, but the hope is that we find some, and then we can link those patients to specific trials targeting those mutations. That's the whole concept of personalized medicine. So if you take home this slide, this is the whole point here. So the team asked me to talk a little bit about genetic testing logistics. They actually sent me some questions ahead of time, so I want to try to address these. So as I said, this could be off a fresh biopsy, or it could be based on your some sample that's stored away in pathology. We actually never throw away the old pathology sample. So even if you had surgery 10 years ago, we can actually retrieve that tumor, and sometimes the DNA is still good enough. So there's a QA process where they look at that to determine if it's okay. There are some nuances in 2 years ago and longer, we used to, for the tumors that were associated with bone, they used to try to decalcify it, take the calcium away from the bone with a very strong acid that would pretty much kill all the DNA. So if the tumor has undergone that process, it's likely that we will have trouble extracting the DNA. We've switched to more gentle techniques, recognizing that this is a key feature of our clinical trial management that are easier. So that's less of an issue now. But anyway, the tumor is sent to either a company or a lab to these complex machines that do the genetic analysis and can pull down which mutations are there. So this can be done through commercial testing or through academics. So I just listed a handful here, but there are a number of different companies that can do this for us. So some of them are just a couple blocks down the street, but the idea here for them is that they're for-profit companies that have expertise in this. They will analyze the samples, report it back to the clinicians. They also partner with pharmaceutical industries. So if you're running a clinical trial as a biotech or a pharma, a lot of these companies will work with them to help them do their DNA analysis. And then the other route is through academics. So many hospitals, many academic institutions actually have their own sequencing capabilities. So Mass General is part of Partners, so is Dana-Farber, but we both have our own systems for doing genetic testing. Johns Hopkins has their own, as well as Sloan Kettering. And then there are clinical trials, national clinical trials, such as this trial called the MATCH trial through the National Cancer Institute that has sequencing built into that. So if you're a participant in that trial, they will do the genetic testing ahead of time. So there's many different routes to do this. There's some subtle differences between the two. So we think there's probably somewhere around 2,000 genes that are important for cancer, and of those, maybe we have 100 that we have drugs for. So they all try to cover the regions that we know we have drugs, I would say in general, but some of them cover more genes, and whether that's important or not, we just don't know yet. Some will cover an entire gene, so a huge area. Some look at a very narrow, what we call a hotspot, where many mutations occur in many different cancers for which we have drugs for. So there's a lot of nuance there, and you can very easily discuss this with your doc or with me about what might be best for you. Okay, so when I send this test, I will get back a report that covers all these genes. So this is the gene list from Foundation Medicine. I don't have any stock in them, I don't work for them, I just happen to use them sometimes. And they give you a report of all these different genes. They have funny names that are abbreviated there. And they'll tell you what's abnormal in your patient sample. So I would say in almost all patients, unless there's a technical issue, we find something, but whether we can act on that or not remains to be determined. It's usually the minority of patients where that can happen. But what we're hoping is that we find some mutation that can link this patient to a particular trial that we're interested in. Okay? All right, so let's see, I got about 7 minutes left. So let me just answer some logistical questions. So when should the testing be sent? So I get this question a lot. So it's early days for genetic testing within Chordoma, and we still don't know exactly what the role of this will play in our patients. So my feeling is that we need to wait to send this genetic testing for individual patients until a patient's ready for a trial. So what do I mean by that? So at the first time a patient presents to the clinic, if it's a localized tumor that Dr. Hornacek's going to take out after some radiation, I would not recommend genetic testing at that time. I would instead wait until either a tumor has come back or it's spread to other areas and that patient is ready for a trial. So why do I say that? So there are high costs that are built into this, and this testing can cost upwards of $7,000, and sometimes that's not covered by insurance. If it's run through academics, there's different ways to pay for it, but we don't want to put that burden onto patients without a clear goal of what that's going to achieve. The second part is that the technology is evolving. So, even 2 years ago or 3 years ago, we were looking at a very different set of genes for our sarcoma patients than we are now. So, if you don't need this testing right now and you can wait 2 years, I can guarantee you that we're going to be testing a different set of genes 2 years from now than we are today. So it's worth waiting until we actually need to consider that testing to match patients with trials, if that makes sense. And then one of the hard things about chordoma is that we're not seeing these classic mutations that we see that drive other cancers like lung cancer, melanoma, breast cancer. And we heard a lot in the last couple of days from some of our scientists who study this field that it's a relatively quiet genome. So I think for now, if we can wait, it's probably better unless somebody's exactly looking for a trial, and that's probably the time to do it. And then just a little soapbox thing is please, unless you're independently wealthy, try not to pay for this yourself. There are many routes to get support for this. It's a high cost, and the burden really shouldn't be put on the patient, in my mind. There are many of the commercial groups have financial aid packages that you can apply for, and there are academic routes to get some of this testing as well. So. Okay, so we got about 4 minutes left. So let me just take you through how I think about bringing a chordoma patient to clinical trials. So the first question I always want to confirm with my pathologist is what's the subtype of chordoma. We've heard about the pediatric or poorly differentiated type. We've heard about conventional chordoma. They're managed very differently. I'll talk about that in a second. What's the rate of growth? So many of you have probably heard or recognized from yourselves that chordoma can sometimes grow very slowly, a few millimeters every 3 months. It might not be the best time to jump on some trial that we haven't, may or may not work, that may have side effects when your tumor is not really growing. It's probably better to wait until we have, the best trial we can find for you if you're not having any symptoms. So that's a discussion to be had in person with the patient. The prior therapies matter. So for some of these immunotherapies that are coming down the road, they might not allow you to participate if you've had other immunotherapies. There are practical matters such as a visit schedule and distance travel. So, we see patients from all over the world in our clinic, and some of these trials, particularly in my phase 1 group, require multiple visits a week. So it could, they might in the first week require 4 visits, 2 visits the next week, and then weekly visits thereafter. It might not be practical if you're traveling from a long distance. We actually, we've recognized that, and we have it at our hospital, and I know they have this at the NCI also, is built-in support networks for that. So if somebody is traveling, there are applications that people can apply for to help with plane tickets and hotel costs. Okay, and then obviously other medical problems are a factor. So if somebody has some other serious problem, like their kidneys aren't working, that could affect eligibility. And then unfortunately, insurance is still an issue. So these trials are funny. So the way clinical trials work in the United States is that typically the routine standard of care costs, things like scans, laboratory tests, those are covered by your insurance company, but things that are outside of that, such as the drug, extra blood tests, are covered by the drug company. So we still rely on insurance for a big aspect of supporting those trials, and there are some insurance companies that will refuse to let patients participate in phase 1 studies. So it does become an issue, at least at our site. Okay, so let's, in the last 2 minutes here, let's just go through the logistics. So when I think about poorly differentiated chordoma, the most important question that I'm asking my head is, has this patient received appropriate chemotherapy? So through conversations with colleagues, my own experience, and then an interesting case report that came out, we're learning that the poorly differentiated chordoma subtype is very sensitive to certain chemotherapy. And so those patients really should not participate in a clinical trial until they've been treated with, at least given a shot at traditional chemotherapy. After that, I don't have time to talk about it today, but there's this drug called tazemetostat that works in a pathway that is altered in poorly differentiated chordoma. We have no idea if this is going to work in this disease. I think there's been 1 patient treated on the clinical trial so far, but if somebody was looking for a trial, that would be my first choice for the poorly differentiated type. So for the other 97% of chordomas, this is my line of thought. So the first priority is to enroll to the open chordoma-specific trials. And it doesn't matter to me if it's not at my site. I'd like to try to make it happen where we can send people to wherever the chordoma studies are open. We only have a limited number of patients, and we can't have competing studies with competing eligibility. And my own personal bias here is that we need to finish the trials we open for that are specific for chordoma first before we open others. So if they have a localized tumor or insignificant lung metastases, I'm going to send it to Dr. Hiri theory if it's reasonable for the vaccine study. We recently had a proton beam study that I think is closed. Is that right, Tom, or is it still open?
We have about 8 more slots.
8 more slots. Okay, so that's another study for radiation-only patients. And then we recently finished a study that included this drug called nilotinib plus radiation, which is similar to Gleevec, which you may have heard about. And we're hoping to publish that study this year. So priority 1 is the open studies. Priority 2 is trying to use this personalized medicine approach to match patients to either phase 1 studies that I have or that other institutions have, or to put them into what we call these basket studies, which are basically phase 2 studies. So the drug dose is already established, but now we're looking at all different kinds of cancers who have a similar mutation. So I try to route patients into one of those two. Thirdly, one of my interests in the phase 1 group is novel agents, so not necessarily this traditional pathway of trying to find some molecule that blocks a certain protein pathway, but rather sort of creative ideas that can try to attack cancers, be it epigenetics, metabolism, new ways to deliver drugs. So we have a number of those studies that are open, and we'll look for patients for that. And then lastly, there's this concept of off-label use you may have heard about. So what's that mean? So when a drug is FDA approved, it's given a label, and it says this is approved for breast cancer, it's approved for lung cancer, you can only use it in these diseases, insurance will only pay for it in these diseases. Well, for orphan diseases where we don't have that luxury to have these big studies, we sometimes pick up mutations that that drug that's approved for breast cancer may be relevant for chordoma. We just don't know. So we can argue with insurance companies to try to get access to that drug to bring it to our patients. So in order, open trials first, using personalized medicine to look for genetic targets, novel phase 1 agents, and then lastly, the off-label use. So obviously, this goes without saying, but we're not going to find a cure for this disease without collaboration between our scientific groups, our drug developers, clinicians, and patients like you. And we got to keep fighting. We've made a lot of progress. I think we're going to get there soon. So we can wrap up there. Thank you.